stuff?
Hey man, don't worry, we're almost done with this deadly virus. Its job is to go straight into the target cell. No. It bypasses the cell membrane because it has an outer coating, a protein. It then heads into the nucleus and modifies the cell's DNA to create more viruses. This instead of healthy cells. In humans, the virus targets the part of the brain, and that is responsible for thinking and memories, brings them down to their natural survival instincts.
Alright, man. Sounds good. Let me know when it's done.
Will do, man. Have a good one.
You got my virus?
Yeah, man. Here it is. I just need my money.
Here you go. It's all in here. Look, here's all the money. Alright?
Thanks, man. Nice working for you.
Yeah. Just one thing. Alright? Nobody can find out about this. Understood?
Yeah.
If someone finds out, you're dead, man.
Yeah, man. No worries. It's safe with me.
Alright, the lawyer's gonna forget what his case is all about, making me win by default.
Yeah, he is.
Hey guys, I was just told by a scientist that we need to go in and figure out what if the zombie person had some sort of infectious disease.
Wait, what do you mean going?
I mean, like, literally, go into the cell and find out if there's anything potentially hazardous, like a virus or a bacteria.
That seems very interesting. How exactly do we get in the cell though? I know the cell has a plasma membrane, also known as the cell membrane, does not accept just any organic compound. I know the cell membrane and the cell wall act like a filter, only allowing a small non-polar or hydrophobic molecules to freely pass through the phospholipid bilayer, which is part of the cell membrane.
Yes, that is true. You guys will be shrunk down beyond real physical limits, breaking the laws of physics. You'll be size of macromolecules, meaning you'll be the size of a very large molecule.
How are we going without destroying the cell though?
Well, that is easy. You will go in being surrounded by a solution, and I will make sure your shape's compatible with the protein so you can enter using facilitated diffusion, which means that the reason why you aren't using a hypertonic and hypotonic solution is that they would not help, since the only way to enter a cell is through the small passageways, no matter the concentration. And you might ask, why aren't we using osmosis, diffusion, or active transport? Well, because osmosis only works with very small molecules, and it's when a solution goes from a lower concentration to a higher concentration. Diffusion is when the solution even out on both sides of the membrane. Active transport is when the cell requires energy to allow something through.
I just said what because I really don't understand none of this, and I just heard the story behind all this.
Well, I could have told you that. I'm a scientist.
Well, that's right. And I guess you'll be the one leading the expedition.
Well, I guess. Why not? I'm the most qualified, right?
Okay, now that we're inside the cell, where should we look? The virus could be anywhere.
Well, it could possibly take the recycling centers to distribute itself. I don't know.
What do you think? We should go. Alright, let's go.
Alright, sounds like a plan. Ah, the lysosomes. Did you know there are two kinds of recycling centers? There are the lysosomes who break down pretty much any material with using enzymes, and then there are the peroxisomes that work with the mitochondria to break down materials.
Interesting. There doesn't seem to be the virus anywhere around here. Maybe let's look in the mitochondria. You said there's reactions there, right?
That's right.
This seems like a good start. Let's see how it goes. We just got here through transport proteins, also known as vesicles. We now stand here at the mitochondria. I like this place, dude. It looks flat. But we rode the cytoscope, I think. That's what Mike, the what's it called, scientist said. And it's all along this place known as cytoplasm. Seems nice.
Yeah, that's what I said, but don't be trying to take my job, alright? It's my job. I'm the scientist here. there's still no sign of it. Where could it be?
I don't know, but I'm getting really hungry. We need to find somewhere to eat.
Maybe. Well, we could go to the Golgi apparatus, and then from there we go on to the smooth endoplastic reticulum and get something to eat.
Sounds good.
Actually, I'm hungry too. Let's just go straight up to the smoothie yard and get some lipids and carbs.
Yeah, better not taste like cafeteria food, dude. I want some good food in here.
Yeah, don't worry about it, man. We got some good food.
Well, let's go eat. We just came back from the smoothie yard. We have brought some lipids and some carbohydrates. Is that correct?
Yes, that is correct. The smoothie yard produces lipids and carbohydrates like glucose.
It seems like way better than cafeteria food, like no joke. Unhealthy, but tastes better.
I want to taste the liquid. Did you know that these monosaccharides can bond together to create disaccharides? This happens through a process called water dehydration synthesis, my bad. That is when a water molecule is lost during the process of bonding. If this happens more than once, it is called a polysaccharide.
So, this happens with lipids too, right? So what happens what is it called when they break up?
Well, when they break up, it's called hydrolysis. So, in lipids, you add water and you would get cholesterol and fatty acids when it breaks down.
I did not know that. You learn something new every day.
Yep.
Now I'm gonna get my tea.
So this is the roof yard, right? There's no sign of him here. I wonder where he could be at. But it's interesting. What do they do in this place?
Well, actually, this place is the rough ER. It is called rough because of all the ribosomes that line it. The ribosomes are actually made in the nucleus, or nucleolus, but this place is responsible for creating proteins out of the amino acids. Now, from here, all of the proteins are shipped to the Golgi apparatus to be prepared for shipment in other places around the cell.
That's a lot of information. We need to find this virus now. So this is the Golgi apparatus where they package and ship all things out into the cell.
Yep, that's right. Doesn't look that busy though.
Yeah, it's empty.
Yeah. So let's go to the rough ER. And by the way, this place is where they ship and package everything, all the nutrients where they need to go.
Easy.
Alright, now that we crossed the nuclear membrane, we're in the nucleus, where all the nucleic acids like DNA and RNA are stored, which are made out of a nitrogenous base, a sugar, and a phosphate group.
Well, let's look for this virus first. So, like, good thing the chromatin keeps everything well organized and compact.
Is this it?
Yeah. It hasn't even reached the nucleus yet. Well, not the DNA yet.
Oh, good thing we found it before it did any damage. Let's take it.
Good. I'm so happy the test is finally over.
Yeah, good thing the case is over and we have positive results.
So how did we get out of here?
Well, we came in through endocytosis, which is when a cell takes us in. So we're gonna leave through exocytosis, which is when the cell takes us out.
Good thing there was no Cecilia or Flagella. Those legs and those hairs, man, they keep me out.
So what now, do we get paid? Taking out this virus? We saved the world. Yeah.
You can stop now.
That wasn't part of the script.
Oh, you guys should have told me.
We just
I was like, okay. Guess who has a job in the elementary school?
Oh.
Oh, are you recording? Oh, sorry.
We get all stuff in there.
It's okay, Megan. They deserve it.
We had 2 lights.
Because when water runs out, that's the only thing left.
Then there wouldn't be iodine. Iodine requires water.
Right after the lunch.
Cool, it's recording now.
Dang. You're gonna be like, oh, you're gonna be like, so this is the Gojo press where everything is shipped. Mike's gonna be like, yeah, it seems like it's not that busy. It's all empty. Let's go to a rough yard and to a rough yard. By the way, this place ships proteins where they are needed after sorting them. Right. This place ships everything. All the nutrients. You can't even see on the camera. You're standing right here.
Yeah, but if I'm like standing here, there.
No, you're sitting down.
Oh, I'm sitting down?
Yeah. Okay. Wait.
I got it.
Sit down, Mike. See if
Mike, tackle
see
me.
if it looks.
Tackle me as I'm coming in.
Like sit down.
No, you can do it hard. You can do it hard.
No, just go. I won't let you hurt.
All right, Mike. Sit down. See if you can see it. All right.
Coming up on the prey.
Can I see the paper? Nope. No paper. All right, Danny, you ready? You're gonna be right next to Mike. I'm being.
Can I be in the chair?
Wait, can you
I
see
look
me?
more badass in the chair.
No, because he has to read the long line.
I'm gonna go.
I'm gonna be behind you guys. Mike, you're gonna cut out the first part because I'm gonna be clicking it. All right? Wait. Wait.
We have retrieved.
All right.
We have.
Don't shake the table. Careful.
Shut.
Okay, okay.
Take two. Go.
Take 5. Take 5. We just came back from the smoothie yard. We have brought some lipids and some carbohydrates. Is that right?
That is correct. We just came back from Miss G.
Making her always interrupting our scenes.
Seriously.
God.
Alright.
Alright.
Pick up.
We just came back from the.
You need some of these? Well, not right now, but in a bit. Seems like. Where's my phone? Right there?
Yeah.
Is it?
Awesome. No, it doesn't.
Alright. So you're gonna be like, you know these mounts? Gonna be like, are you able to bond using hydration synthesis?